Thank you. So to start off with this morning, I'll be talking about two particular genes that our lab has been doing work on for a number of years. The first being OPRN1 in relationship to alcohol use disorders, and secondly, CHRNA4 in relationship to tobacco, given today's focus on nicotine. And then I'll be talking about individuals with this particular comorbidity. I'll briefly touch on environmental determinants, specifically family history and alcohol, and the future direction. So why is this important? Dr. Fowler really beat me to the punch this morning in that our substance use disorders are both highly heritable and highly influenced by the environment. And NIH has really developed a push to identify these mechanisms, to identify why individuals are more susceptible for substance use disorder. And having better understanding of mechanisms will hopefully in turn allow us to better identify and improve our interventions and treatment, our quality of care, and most importantly, our outcomes with our patients. Another push by NIH, in particular NIDA and NIAAA, is the development of personalized medicine, which will allow us to identify genetic mechanisms that predict treatment response, and therefore we can improve treatment efficacy by putting people in the medications and in the intervention conditions that they're most likely to respond to. So just to touch on heritability and why the genetic influence is so important, this table really shows that of the various substances, various studies have shown them all to be highly heritable. The top three are, of course, are legal substances, alcohol, tobacco, and caffeine, with estimates ranging from about 40% to upwards of 70% heritability rates, again, depending on what study and what sample you're looking at. On the bottom half, you can see several examples of illicit substances, marijuana, cocaine, and opioids. Opioids are a little bit lower than the other two, but again, showing somewhere between 40 and 70% heritability rates. One of the genes that's been of real particular interest in the past 15 years or so is OPRN1 in relationship to alcohol use disorders. And of course, alcohol is highly prevalent in the U.S. Data from SAMHSA last year shows about over half of the U.S. population reporting current alcohol use, and again, somewhere between 7 and 10% needing for an alcohol use disorder. The gene I'll be talking about today is the A11AG polymorphism, OPRN1. The majority of individuals are major A allele homozygotes. You might see this denoted as ASN40. And then a particular clinical interest are individuals who have the minor G allele, which is denoted ASP40. And individuals of European descent, about 15 to 20% of individuals have this particular gene expression. It's lower in African Americans, more around 5%, and it can be as high as 50% in individuals of Asian descent. OPRN1 became of interest because of the opioidergic system's likely influence on drug-induced feelings of analgesia, withdrawal, and euphoria. So why are we so interested in these G allele pairs? There's some evidence that they may have more potent striatal dopamine release, again indicative of alcohol's influence on the hedonic reward system. These individuals in laboratory studies have been shown to have greater alcohol craving, alcohol-induced stimulation, and greater alcohol-induced vigor in the lab, showing that they have just different subjective impacts of alcohol. They also exhibit greater cue-induced craving, meaning if this person was at the bar, even though they were on treatment and been abstinent for a while, their cue-induced craving of just being around alcohol is going to be greater than those A allele homozygotes. This is some of the earlier work on this gene published by my mentor, Dr. Ray, in 2004. They brought in 38 heavy drinking college students, half of whom were the major A allele homozygotes, and half of whom had that G expression. They gave everyone an infusion of alcohol, and at 3 time points asked them to rate their subjective feelings of intoxication. So at point of 2 breath alcohol, point of 4, and then at point of 6. As you can see from this graph, across those VACs, the G allele carriers expressed greater subjective intoxication. And a similar pattern was found for alcohol-induced stimulation. Now, college samples are wonderful. They inform us a lot about our treatments and about clinical response. However, to replicate this finding, once Laura got to UCLA, we completed another study in our lab looking at a community sample of heavy drinkers. So we recruited 43 individuals from the greater LA community, again, half of whom had the major A allele homozygote expression, half of whom had a G allele expression. They came in for 2 sessions. In one session, they got a saline infusion, and then in another session they got an alcohol infusion. And again, we took measures at these time points of different BACs. As you can see on the left from the allele homozygote chart, pretty stable stimulation, self-reported stimulation report across placebo, a little bit higher for alcohol, nothing surprising there. However, what becomes interesting is when we start looking at these allele carriers. As you can see in relation to placebo, a relatively stable report of subjective stimulation. However, when given alcohol, they report greater stimulation as compared to placebo and as compared to those allele homozygotes, again showing that hedonic response to alcohol. So why does this matter clinically? One of the main medications for alcohol is, of course, naltrexone, which is a mu opioid receptor antagonist. It's been shown to be the frontline alcohol pharmacotherapy, as demonstrated by the combined study published in 2006. Carriers of at least one copy of that minor G allele were shown to have better clinical response to naltrexone from the combined study data, and a meta-analysis published a couple of years ago also showed that those carriers had lower relapse rates in alcohol-dependent individuals. I do want to acknowledge that this data is mixed, however. A couple of studies have shown absolutely no effects of naltrexone by OPRM1 and some of the other OPR genes, including OPRD1 and OPRK1, have shown no treatment effects, no treatment interactions. This one is pretty much hot off the presses. One of the most recent clinical trials published by Dr. Oslin and colleagues at the University of Pennsylvania, which was published a couple of months ago. They recruited over 220 alcohol-dependent individuals and prospectively genotyped everyone and then randomized them to either receive naltrexone or placebo for 12 weeks in combination with medication management. This is one of the first prospective genotyping studies to date, and you're all very excited about it. However, as you can see from this graph, the treatment groups did not separate out very well. The good news is that everyone stopped, or at least reduced their drinking somewhat. However, there was no gene interaction when controlling for slight differences in baseline rates of heavy drinking. However, there is ongoing work that we're still excited about. We're not throwing the baby out with the bathwater. There's another prospective genotyping study currently being conducted and actually just right now wrapping up at the Medical University of South Carolina that is again investigating those G allele carriers' response to naltrexone. Interestingly, the study is incorporating an imaging component with those G allele carriers receiving naltrexone to look at damping to induce brain response. In our lab, currently, we have an ongoing clinical trial looking at naltrexone, specifically in Asian Americans. As I mentioned, there's a higher prevalence of that gene in Asian Americans, and we're wondering if naltrexone, therefore, might be a better treatment option for that group of drinkers. Turning to tobacco, one of the genes of interest has been CHRNA4. As has already been noted today, although smoking rates have decreased over the past few decades, they're holding pretty steady around 20%. We haven't really been able to break that barrier. So developing better medications and treatment options is, of course, of interest. This particular gene encodes for the alpha-4 nicotine acetylcholine receptor subunit protein, and this has been shown to play a role in the reinforcing effects of nicotine. Variants of this gene are associated with mood disturbance and greater nicotine withdrawal. This particular SNP, RS2236196, is also important in that minor allele carriers of the gene have reported greater nicotine high, which is physical effects, cognitive effects, than major allele homozygotes, showing that they might be a bit more impacted by initial nicotine administration. In one large German sample, this particular SNP is also associated with higher FTND scores, a measure of nicotine dependent severity, and greater sensitivity to withdrawal. Varenicline is currently the frontline pharmacotherapy for smoking cessation. It's a partial agonist of the alpha-4 beta-2 nicotine acetylcholine receptor. It has been more effective than nicotine replacement therapy, bupropion, and placebo, and about 25% of those treated with varenicline are able to successfully maintain abstinence at 6-month follow-up. However, this number is still not great. We're doing okay, we're not doing great in terms of helping these people truly stay off cigarettes and other nicotine products. CHRNA4 expression is associated with having better odds of abstinence between weeks 9 and 12 when treated with varenicline in one clinical trial. However, this finding didn't hold at 1-year follow-up. And of course, I don't need to tell this crowd, these two substances and behaviors run hand in hand with each other and frequently coincide, with close to a quarter of those with nicotine dependence also the main criteria for alcohol use disorder. These individuals are also harder to treat, in part because these behaviors coincide, with individuals being 5 times more likely to lapse on days when they're drinking than days when they're not drinking to pick up nicotine again. So how do we improve care? One way is to identify medications that improve an effect's response to both alcohol and nicotine, and again, the personalized medicine approach I mentioned earlier to identify genetic mechanisms that improve treatment efficacy. Now, there has been some evidence suggesting that varenicline is efficacious for alcohol use disorders, some evidence that naltrexone can impact smoking behaviors, and some evidence that OPRM1 may play a role in nicotine use. I just briefly want to touch on a study that was completed in our lab at UCLA. We received funding from the tobacco-related disease research program here in California to recruit heavy drinking smokers. We recruited 130 participants and randomized them to one of 4 medication conditions, one being the combination of varenicline with naltrexone, naltrexone alone, varenicline alone, or placebo. Everyone had to report smoking at least 10 cigarettes per day. Be heavy drinkers according to NIAAA criteria. I'll be discussing the genetic analyses. However, the main outcome paper was published last year in Psychopharmacology, which showed that the combination of medications was superior to placebo and in some cases monotherapy at reducing cigarette craving, cigarette high, and ad-lib substance consumption during the medication period. For my part of the study, I looked at the 67 Caucasian participants and looked at their genotyping analysis. They completed the timeline follow-up and 2 measures of craving, one for cigarettes and one for alcohol. Our first result was a significant gene-by-medication interaction. This was taken at the baseline when they came back in for laboratory testing after 9 days of medication. Our post-hoc pairwise comparison showed that this group over here, who received varenicline, who were minor allele carriers, had less alcohol craving than their major T allele homozygotes also treated with varenicline. And secondly, we observed a similar trend level pattern. This is for cigarette craving. And again, the minor C allele carriers treated with varenicline reported less cigarette craving than their T allele homozygote counterparts also treated with varenicline. And while these results are incredibly preliminary in nature, we are excited about them. We find them promising, and we're actually starting a larger, longer clinical trial next month to be able to better examine these gene by medication interactions. Just to wrap up, I was going to hit a little bit on environmental determinants. They've already been mentioned today. There are, of course, a lot of unique and shared environmental factors that contribute to the development of the substance use disorder, including life events, parenting, family environment. But data from Ken Kendler's group has really posited that environmental factors exert more influence at younger ages versus genetic influence becomes more important as one grows up and moves into adulthood. Of particular interest to me is the family history component, as it's difficult to disentangle what's related to genetics and what's related to one's family environment. For alcohol use disorders in particular, environmental influence can range from 40 to about 60%. And was mentioned earlier today, family history of an alcohol use disorder is a known predictor. One theory is that this is because children are drinking at an earlier age, and there has been data to suggest that earlier ages of initiation are predictive of development of a disorder. However, I do want to add the caveat that context of drinking initiation does matter. We've done some studies showing that actually drinking initiation beginning in the home at a family event is more protective than when individuals initiate drinking with their peers. Also, environmental influences, such as is it acceptable to drink heavily in the home? Are there relationship stressors? Are there socioeconomic concerns? And finally, personality factors. There's some evidence that children of alcoholics have higher impulsivity and novelty seeking and lower agreeableness. However, of course, personality is highly heritable as well as strong genetic influences. I do want to note that there has been some evidence that G allele carriers are more likely to report a family history of alcohol use disorders, again alluding to the fact that it's very hard to disentangle this relationship. And finally, the treatment implication for this evidence there have been several studies to replicate the finding that naltrexone is more efficacious for those with a family history. What hasn't been examined is if there's a genetic moderator of this relationship. This is my final piece of data from a Monterosso paper in 2001. As you can see, the individuals all the way to the right were reporting the highest level of alcohol problems within their family, as determined by the ASI interview. When treated with naltrexone, they reported significantly less days of heavy drinking compared to their counterparts who received placebo, but who also had a severe family history. And finally, of course, there's more work needed. We need more gene by environment studies to disentangle this relationship between family history and genetics. Our field is getting more advanced at looking at gene interactions, such as work done by OPRM1 and DAT1 from both our lab and USC. We need more pharmacotherapy development and pharmacogenetic exploration. And finally, novel methodologies, such as translational work and multidisciplinary teams, and the use of new methodologies, such as fMRI. With that, I'd like to thank NIDA and NIAAA, particularly the ISAP 232 that's been so supportive of me, my mentor, Dr. Ray, and our whole UCLA Addictions Lab.
Well, time for one question, and then we have a couple of distinguished guests to introduce over here.
Just a clarifying question. Asian Americans have
Close to. It's not that there's necessarily a higher rate of alcoholism, it's that you might be more susceptible to it and that you might experience greater high, greater craving, the subjective effect might be different. And it's that 50% number varies again from study to study, but up to 50%, depending on which racial group you're looking at, can have that gene expression. Is that
yes.
Thank you, Emily. Great talk, really exciting work, and we hope to hear more from you. I have a couple of distinguished visitors join us today. I'd like them to give a brief university welcome. Provost Steve Michael first.
I'm sorry, I was supposed to be here very early on to welcome you to our group university, but as you can see, there's a big tent out there, and we do opening the new university hospital over there. So we just finished some of the events I'm over here because the president of the Senate said, you are here, and I'm happy to be here to welcome you. So welcome to Charles University. The truth is that there's nothing that defines you as a scholar more than commitment to excellence, and there's nothing that defines excellence more than doing research work. All right, because this is what makes you critique the status quo and expanding new revelations. All right, and taking us to the next level of excellence. So I want to thank you for the work that you are doing, and we look forward to actually benefiting and learning more. Thank you very much.
Thank you. Madama?
Good morning and good afternoon. My name is Jay Bakama. I'm Vice President of Research. Again, I'd like to welcome you all. Just as the provost mentioned, some of us were tied up this morning as a result of the opening of the new hospital. And as I was looking across the hospital and this particular program, and I've had the privilege to see this program now in its 10th year of its celebration and how far it has come. But I'm sure you heard we have a phenomenal opportunity across the road in terms of our mental health programs. And a lot of work that each and every one of you doing here provides a fantastic opportunity for collaboration, clinical trials, what the Los Angeles County has invested across the road. Again, I congratulate you. I know a lot of the students in this room get a chance to interact with them and treatment. Congratulations. And welcome everybody.
Thank you, Jay. Thank you, Dr. Badama.